The molecule movement across a semi-permeable membrane simulation is an interactive educational tool designed to help students understand the principles of diffusion, osmosis, and selective permeability. This simulation allows users to study the movement of molecules across a semi-permeable membrane, providing insights into how different sized solutes interact with the membrane. By experimenting with three solutes of varying sizes, students can observe the selective nature of the membrane and the factors that influence molecular movement. Understanding semi-permeable membranes. A semi-permeable membrane is a barrier that allows certain molecules to pass through while blocking others based on their size, charge, or other properties. In biological systems, cell membranes are semi-permeable, regulating the movement of substances into and out of cells. The simulation models a semi-permeable membrane that separates two sides of a beaker, with one side containing a solution of solutes and the other side containing pure water. Key concepts explored in this simulation. 1. Diffusion. Diffusion is the movement of molecules from an area of high concentration to an area of low concentration. In the simulation, students can observe how small and medium-sized molecules diffuse across the membrane, driven by the concentration gradient. 2. Osmosis. Osmosis is the movement of water molecules across a semi-permeable membrane from an area of low solute concentration to an area of high solute concentration. The simulation allows students to observe how water molecules move across the membrane in response to the presence of solutes. 3. Selective permeability. The membrane in the simulation is selectively permeable, allowing small and medium-sized molecules to pass through while blocking large molecules. This selective permeability is a crucial concept in biology, as it enables cells to control the entry and exit of substances. Simulation setup. This simulation is carried out in a virtual beaker, divided into two compartments by a semi-permeable membrane. Users can select from three solutes of varying sizes: small, medium, and large. The membrane is impermeable to the large molecule and permeable to the medium and small molecules. By adding different combinations of solutes to one side of the beaker, students can observe how the molecules interact with the membrane and move across it. Simulation scenarios. 1. Small molecules. When small molecules are added to one side of the beaker, they quickly diffuse across the membrane to the other side. This scenario demonstrates the principle of diffusion, where molecules move from a high concentration to a low concentration until equilibrium is reached. 2. Medium molecules. Medium-sized molecules also diffuse across the membrane, but at a slower rate compared to small molecules. This scenario highlights the effect of molecular size on diffusion rates and the selective nature of the membrane. 3. Large molecules. Large molecules are unable to pass through the membrane due to their size. This scenario demonstrates the concept of selective permeability, where the membrane allows certain molecules to pass while blocking others. 4. Combination of solutes. By adding a combination of small, medium, and large molecules to one side of the beaker, students can observe the differential movement of the solutes. Small and medium molecules diffuse across the membrane, while large molecules remain on one side. This scenario provides a comprehensive understanding of how different sized solutes interact with a semi-permeable membrane. This simulation also allows students to study the process of osmosis. By adding solutes to one side of the beaker, students can observe how water molecules move across the membrane in response to the concentration gradient. In a solution with a high concentration of solutes, water molecules move into the solution to equalize the concentration, demonstrating the principle of osmosis. Benefits of this simulation. 1. Interactive learning. The simulation provides an interactive platform where students can actively engage with the concepts of diffusion, osmosis, and selective permeability. By adding different solutes and observing their movement, students can gain a deeper understanding of these fundamental principles. 2. Visual representation. The visual nature of the simulation makes abstract concepts more tangible. Students can observe the movement of molecules in real time, helping them to grasp complex ideas more easily. 3. Immediate feedback. The simulation offers immediate feedback, allowing students to see the results of their experiments instantly. This helps correct misunderstandings quickly and reinforces learning through trial and error. 4. Flexibility. The simulation is flexible, allowing students to explore a wide range of scenarios. Whether investigating the movement of small, medium, or large molecules, the simulation provides a comprehensive learning experience. The molecule movement across a semi-permeable membrane simulation is a valuable tool for teaching and learning about the principles of diffusion, osmosis, and selective permeability. By providing an interactive, visual, and flexible platform, it helps students understand the fundamental principles of molecular movement in biological systems. Whether used in a classroom setting or for individual study, this simulation can significantly enhance the learning experience and build a strong foundation in cell biology and chemistry. Through exploration and experimentation, students can gain a deeper appreciation for the complex and fascinating interactions that occur across semi-permeable membranes.